Hi, this is Miss Litton, and this is a Honors Bio Chapter 25 review, and this is about control of gene expression and cancer. And the first thing I want to ask you is, where are you? It's a very small room in here today, and I guess you are working towards your other finals, but it's best if you can come to the review instead of watching them from afar. So the first thing I want to talk to you about in this is that and I'm going to hit all the main points here, is that the DNA in every cell is exactly the what? Same. Yeah. In that organism, all the DNA is the same, yet different aspects of it are expressed in different cells. So we want to understand, how does that occur? When we talk about, where is everybody today? Okay, it's your fault, I'm sure. When we talk about gene regulation, it's going to be different whether we're talking about it in prokaryotes and in eukaryotes. She wants you to share with her. Whether you're talking about it in prokaryotes or eukaryotes. And because which one has a nucleus? Eukaryotes, yeah. So there are steps. We know that when you go from DNA to RNA, what is that called? DNA to RNA? Transcription, good. And then using that RNA to build a protein is called translation. So there are steps all along the way in this process. And we can use this diagram just to try something new here to review that. So if we're looking at the DNA, if it's even available, where is your stuff?
I don't want to disturb you.
Okay. If you're looking at DNA, if it's even available, that is when you're talking about chromatin or chromatin. Is that DNA even available? If you're talking about, can RNA polymerase get on the promoter, that kind of control is called what? Transcriptional control. If you're modifying the RNA, like a cap, a tail, right? The ability to leave the nucleus, cutting out the what? Introns. Okay, that part right here, cap tail, cutting out the introns, that's called post-transcriptional, right? If then you're talking about the RNA getting on a ribosome, the mRNA being on a ribosome so that you can build that protein, that's translational control. If you are folding that protein in some way or modifying it, that is post-translational. That is for eukaryotic cells because they have a what? Nucleus. Okay? So if we numbered it, this would be 1, this would be 2, this would be 3, this would be 4, and this one would be 5. Okay? Any questions or concerns about that part of it? We just said, and it could happen all along the way. So when you talk about how different genes are expressed at different times, different locations, you call that differential gene expression. And we started off just going with the simplest of all, and that was looking at prokaryotes and how their genes are regulated. And in order to understand that, we needed to understand what a promoter is. What is a promoter?
Where RNA initially binds.
Yep, perfect. Where RNA polymerase binds. And what about an operator? What can bind to an operator? Repressor. Yeah. So a repressor can bind there. If that repressor binds there, then there's going to be, you won't have any of these genes, these structural genes transcribed or translated. However, if the repressor is absent, then those genes can be transcribed and translated. Who made this repressor? Where did the information, because a repressor is a protein, who made it? Yeah, the regulator gene. So the regulator gene was transcribed and translated into that repressor, and the repressor could get on the operator. Now, if I want to get, we talked about the lac operon. If I want to get that repressor away, what needs to bind to the repressor to make it go away?
Lactose.
Yes, in this specific example, lactose. Because if we bind lactose, and let me go here. So here was our repressor sitting on our operator, and as a result, RNA polymerase could not get on the promoter. It's not a bad thing because I don't need these genes transcribed and translated because they are genes that code for enzymes that digest lactose. So if there's no lactose present, what do I care? Let's not make those proteins. But like you said, if lactose is present, then lactose will bind to the repressor, and then that will cause the repressor, he can only hang on to one thing, so now the repressor is gone. Are you out of here?
I'm out of here.
Are you doing your therapy?
My therapy? No, I'm just out of here.
Okay, have fun. Okay, so then the RNA polymerase can bind to the promoter, it can chug right along here, and you can make your mRNA. If that mRNA is translated out on a ribosome, it will make a protein. What will that protein do? Digest lactose. So if our lactose starts to get digested, including the one that's binding to the repressor, now it can hang on to the operator again, and that's okay. That's really good feedback because I'm all done digesting lactose, so I don't need to spend any more time turning on that gene and coding for that protein. We talked about the analogy we used is in your home right now, in your bedroom or bathroom, hopefully bathroom, not bedroom, but you have a shower. That currently the shower is what? Off. There's a repressor that's preventing the water from going out of your shower. If you need the water, you will turn a knob which will allow the water to come out. If you need the enzyme to digest lactose, then lactose is the thing that's turning the knob, that's removing the repressor so that the water comes out. Okay, that was with the lac operon. Any clarifying questions on that? Okay, so let's do some questions. Do you want me to go over it with that? Are you feeling okay with it? I can leave those. Okay, so let's do some questions. It's 2000 number. So you put 2000 and then the two numbers. Need your 5th period attendance. Yeah, I wonder who wasn't here today. That is a mystery. Oh, there's 14 of you in here now. Wow. I'd like to know where you are. All right, so what's our answer on this one?
The operator.
Yeah, the repressor binds to the, to the operator, okay? You want to just go through these out loud, and then we can just kind of do it a little bit faster. Name the part of the operon system where the genes are located that code for the repressors. So which one of these things codes for the repressor?
The regulator.
Yes, good job, the regulator. Okay, name the part of the operon system where that codes for the lactose digesting enzymes. Yep, those structural genes, in this case, they are coding to digest lactose. Some other structural genes, it might be glucose, and another one, it might be maltose, right? So it's just in this example, our structural genes were digesting lactose. Okay, name the part of the operon system that will bind to the repressor, making it inactive. Yes, I agree, lactose. Name the part of the operon system where RNA polymerase binds. Promoter. RNA polymerase binds to the promoter. All right, so then we said we're moving on now to the eukaryotic control. And in this part, you want to remember that in some of our cells, certain genes are always on. They're what you would consider housekeeping genes. And then riddle me this. Would those housekeeping genes that are always be on, if I stained them, would they be heterochromatin or euchromatin? Remember that term, heterochromatin and euchromatin? Euchromatin, okay? You are active, okay? The heterochromatin is dark staining, and here I'll show you a picture. And the reason why it's dark staining is it's all coiled and it's all put away. The dark staining, it's all put away so it's easy to stain. Euchromatin is open. You are active. Okay, maybe that would help you. Okay, so if we look in a red blood cell, hemoglobin would be what? Would be euchromatin, and the insulin and the myosin would be what? Heterochromatin, because you wouldn't be needing myosin or insulin in a red blood cell. But in a muscle cell, okay, hemoglobin would be what? Hetero. Insulin would be hetero, and myosin would be what? U. Okay, and in a pancreatic cell, they would have it heterochromatin for the hemoglobin, because you don't need that. Insulin would be euchromatin, and myosin would be heterochromatin, because you don't need that. So certain DNA is tightly coiled and put away, because that cell doesn't do that, and other parts of it are active. That is an example of differential gene expression. Some traits are getting expressed and some not. So then we looked at this and we talked about the levels of control, and this is what we started off with in our review today. We talked about chromatin structure. Is the DNA tightly coiled and put away? If it is, it's heterochromatin, it's not active. Or is it available? It's euchromatin. Okay, that was number one. Number two is transcriptional control. Cannot RNA polymerase get on that promoter? Can it transcribe those genes? 3, cap tail, cut out introns, leave the nucleus. 4, am I making a protein? 5, am I modifying that protein? Same as the very first slide, 1, 2, 3, 4, 5. Yes?
Can you go over transcriptional control?
Oh, I'm going to go over them all.
Oh, okay.
I was just going, here are the 5, just so you could see these are the 5 we talked about in the beginning. Okay? So I would know an example of a Barr body that you could see, and that is in females, we have 2 X chromosomes, and so one of our X's condenses into a Barr body, and the other one not. Why don't males have Barr bodies?
Because we have only one X.
You have only one X, so you can't condense one. I was just making sure I did record it. So you can't condense one, you're gonna leave it open. So yours is always gonna be euchromatin. So you can see, and this is just showing you the nucleus. The rest of the cell, okay, is out here. But in this nucleus, people who study this know that that is the Barr body, okay? And that would mean that this cell then is from a female. And where you can see how that plays out in the phenotype is in calico cats, because calico cats, depending on which X is condensed, if the one with the black coloring is condensed and the other one is not, then it'll be tan in those areas. And if the tan one is condensed, if that's the heterochromatin and the black one is the euchromatin, then you'll see black. But the male calico cats will either be black or they will be what? Tan, because they only have one X. So that's an example of a Barr body. Right? Transcriptional control in prokaryotes had to do with repressors that could be shutting down operons. That is not so in eukaryotic cells. We don't have operons. What we have is various genes where RNA polymerase wants to bind to the promoter, but it needs assistance, always needs assistance binding to the promoter. So if there is no assistance, you do not transcribe those genes, right? If there is assistance, you will. What do you call those assistants? They are grouped as transcription factors, which makes sense because it's all about transcription, right? So if you have the correct transcription factors, which could be multiple, right? I see this is just one transcription factor, but it has 3 parts to it. And not only that, to get it in the right spot, it needs to bind to this activator, which needs to bind to this area of the DNA called the enhancer region. So that means the enhancer has to be available, activator has to be available, all 3 parts of this transcription factor have to be available, and this has to loop around in order to help that RNA polymerase bind. If you don't have all 5 of those components, then you will not transcribe those genes. Now, this is a very simplified version of that process, and if you remember, I told you it can be very complicated, all the chemicals that are working together in order to make it possible for you to transcribe those genes. And this part is true in prokaryotes and in eukaryotes. Transcription is the key point of control. There's control all along the way, but this is the really big one, okay, is transcriptional control. We related this to cell signaling and how, you know, maybe, oh, hello, maybe some, okay, crazy slide. Okay. I don't know why it's doing that, but let's look over here in this little corner right here. So this could be some hormone or something secreted by another cell, and it binds to a what kind of protein? Receptor protein, good. Which then starts the signal transduction pathway, and here's that activator that goes and binds the enhancer, which binds the transcription factor, which gets those genes transcribed. Okay. And then transcription factors are like snipers on a roof, postmen delivering mail, or Boy Scouts being helpful. I would just say they're helping that process along. You know, if this is a lame question, okay, just so we're clear, okay? But I wanted you to see, when I think about snipers on a roof, I'm thinking about like a repressor getting in the way of you transcribing those genes. Okay, transcription factors, they're helping RNA polymerase bind to the what? Promoter. Okay, they're helping. Okay. Then we talked about post-transcriptional control. That's the cap, that's the tail, that's cutting out those introns and just leaving the exons, and then it's how fast do you leave that nucleus. So anything right after following transcription. The processing we need in order to make mature RNA. Cap, tail, cut out the introns before it leaves the nucleus. That whole part is post-transcriptional control. We've already made the RNA, we're just modifying it. Okay? Following that, following post-transcriptional control, we have translational control right here. This is, can the ribosome connect with the mRNA and so that you can make that particular polypeptide? That is translational control, right? Translation, when you're doing translation. And then post-translational control is, once you've built this protein right here, does it need to be modified at all in order to get functional? Do you have to fold it in some way to make it functional? Okay, and that is right here, and that's translational control when you're building the protein on the ribosome, and post-translational control is any kind of folding or modification of it. Okay, and then this is a picture too that kind of shows you, you could have chromatin, transcriptional, this would be post-transcriptional, all the way to getting out. Getting on the ribosome is translational control, and then any kind of way you need to fold that protein would be post-translational control. Okay, and here is a question for you. Take a look at that and decide your answer. It's chapter 15, right? I know I got it ready, so let me just check.
No, transcription. Translation is
Okay, so write on the paper, not on here, okay? And then just put what chapter, what version, or whatever I give you there. Okay, so tell me this one. Name the type of control mRNA getting introns removed. What would that be? So smart. Type of control, three-dimensional folding of a protein. Good, because we've already made our protein. We're just making it in the right shape. Good job. Barr bodies. Chromatin structure, because Barr body is referring to DNA that has condensed the second X in particular. Okay. Ability to bind to a ribosome. Translational, because when we're doing translation, we're taking the mRNA, putting it on a ribosome, and using it to build a protein. Right? Okay. And then, good. Okay, any questions on that before I hit the last topic? Okay, so, and by the way, I'm really proud of you. Okay? I'm really proud of you. Your hair, you're reviewing, that's good. Okay, so then cancer, first of all, you want to know like what's normal. So what's normal is cells stop dividing when they're crowded. That's contact inhibition. Another normal is anchorage dependence. Not only do they stop dividing if they're crowded, they must be attached to something. That's normal, okay? Not normal is continuing to grow even when you shouldn't, not exhibiting contact inhibition, not needing to, losing anchorage dependence altogether. That's not normal. And when that initially happens, it might be just localized and staying in one spot, and you get an abnormal growth right there, a neoplasm, and if it stays in one spot, we're going to call it what? Benign, right? And then you hopefully just go in there and cut it out and remove it. But once it starts to move to other areas, it's no longer a benign tumor, it's malignant. And the process of moving is called when it metastasizes. So if it has the ability to move to other areas, okay, it has now become malignant. And the process of it moving, it's metastasizing, it's spreading. Areas that you would be concerned about is if it gets into your blood vessels or in your lymph nodes, because it can get in your ducts and vessels and get spread to all over your body. So abnormal cells are not listening their cell cycle of when to reproduce and when not to reproduce. They're not doing that correctly. And they have the ability, they're going rogue, they're just moving throughout your body wherever they want to be. So why is that? Their DNA is messed up. If their DNA is mutated, then their mRNA is incorrect. And if their mRNA is incorrect, their proteins and cell signaling is incorrect. So carcinogenesis is the beginnings of cancer. This is not how long you live as a result of contracting that particular cancer. Like I mentioned in class, pancreatic cancer moves very rapidly. Sometimes people just have, 4 to 5 months after they realize they have pancreatic cancer. That's not what I'm talking about. I'm talking about how long does it take for you to actually get the cancer. Most cancers take decades before you actually get it, right? People my age, and they're 50 or older, are the ones who normally get skin cancer because you've had 50 years of exposure to the sun. So it usually takes, it's like you accrue mistake after mistake after mistake in your DNA till it reaches a tipping point and you get that cancer. So that's what carcinogenesis is. Another characteristic, we started going into the characteristics of them, is they're undifferentiated. They don't look like all the other cells in that area. So they're undifferentiated. They have abnormal nuclei and cytoplasm space, and they can have multiple nuclei. So that does not look normal. So they're undifferentiated, they have abnormal nuclei. They don't do apoptosis. Normally cells are not immortal. They'll divide about 50 times. Thank you, sweetheart. They'll divide about 50 times and then that's it. Okay, they won't reproduce anymore, but these cells just keep going. And they're not committing suicide even though they're broken. They lose, they don't have anchorage dependency. They just, they're fine wherever they are, and they don't realize that they're crowded. They just continue to reproduce, so they've lost their contact inhibition. They have the ability to metastasize, to spread to other areas. Okay, and evil of all evil, if they're localized in one spot and just growing abnormally, then they would eventually shut down because they'd run out of nutrients. But these cells have the ability to secrete chemicals to make blood vessels grow towards them, and that's called angiogenesis when that occurs. They're forming new blood vessels to supply them with oxygen and to remove their waste. So why does this happen? We attribute this to two major groups of genes. There's several families underneath that, but there are two big groups, proto-oncogenes and tumor suppressor genes. Okay? If you just have a proto-oncogene that mutates to an oncogene, okay, what an oncogene will do is it'll take this normal, here we have a normal cell, okay? But this one is mutating into a cancerous cell. And it is now wanting to make multiple copies of itself. What tumor suppressor genes do is they code, okay, for enzymes that will cause these cells to commit suicide. So you get rid of the bad cells. And then you should still be okay, even though your proto-oncogene has mutated into an oncogene. But if you do not have functional tumor suppressor genes, it can't get rid of these bad cells, so that's when you get a neoplasm. So it's a combination of both of these. Genes that are in control regions, usually for stimulating the cell cycle. And the example I gave you was RAS, because it's like stepping on the what? Gas, yeah. That one says go cell cycle, which is a bad thing, because you're stepping on the gas and you're promoting the cell cycle, but you'll still be okay as long as you have good what? Brakes. But if your tumor suppressor cells, who are like the brakes, are not there, then as a result of this, this cell will continue and make more of itself. There's no apoptosis, and that would be cancer. So example, RAS is an example of a, would be like a proto-oncogene, oncogene, because it's in a position of control. It tells the cell to reproduce if it receives a signal. In this case, this proto-oncogene has become an oncogene because it doesn't care if there's anything in there to signal it or not. It's just telling the cell to reproduce. Yes?
So both are good intention in the beginning, but then they can't.
Yes, these are genes that are in us that are a normal part of how we need to make more cells, right? You do grow. You're getting taller, okay? That's a normal thing. But you want them to listen to the cell signals. So this is one where this, it's the same area in your DNA. If this black region on this pin was the proto-oncogene, when it goes bad, it's still this right area, but it's coding for the incorrect proteins, which are facilitating the cell cycle. So now we call it an oncogene. It mutated into that. They really are saying oncogenes are bad, but before they were bad, they were good regulators, and so we call them proto. Okay? But they're a part of our normal cycle. Then you have other genes in other areas that code for tumor, and one of those is the p53 gene. It makes bad cells kill themselves, or it makes enzymes that will kill that particular cancerous cell. But they're like, they have a gun to kill, but they're shooting blanks. It's not working. So you can't take out those cells that have, what? Yes, that sounds good. Okay, and that's when you ultimately end up getting cancer. Okay, and we talked about, I don't know if do you want these slides? We talked about that. Are you okay with this? Okay. Then last we talked about, well, how am I going to know if I'm going to have cancer? Well, hopefully you don't have any cancer, but cause of cancer, heredity. That can be one reason why. You can inherit that bad gene from a parent. Okay, so that's one way you could get cancer. And a second way is just what you're exposing yourself to in the environment. This would be if you keep exposing yourself UV rays, UV rays, UV rays, UV rays, I'm probably going to get what kind of cancer? Skin cancer. Now, am I going to pass skin cancer on to my children? No, because it's not in my ovaries, right? So I'm not passing that on. That's something I was exposed to, okay, within my lifetime that caused that particular mutation. Or smoking, or some other chemical in my environment, or I got the HPV virus, or something like that. Okay, and then what do you do to check? Well, you can do standard screening processes like mammograms and pap smears, body scans, or whatever you can go to check. And then if you think Oh, and the other one, tumor marker test, that's taking a sample of your blood to look to see if you have any markers in there, any protein markers that would indicate that you have prostate cancer. And that would be like the PSA count. And then genetic tests. So you could go and have your DNA analyzed to see if you have a bad gene, like the breast cancer gene. Then to confirm any diagnosis, oftentimes they want to take a sample of that tissue. You know, it could be just a cyst that's in your breast and they might just go ahead and remove it, but there's no risk of cancer, or it could be cancer-like cells. So they want to take a little sample of it, biopsy to check it. They might take some sort of scan, anything that could image that area in order to confirm that it is cancerous. And then for your treatments, they're either going to send chemicals throughout your entire body in chemotherapy, okay, or they're going to radiate that area that maybe is cancer, or they'll just cut it out. And those are the typical methods. There are some other immunotherapies they're looking at. The future is bright for some of these to prevent blood vessels from growing towards it, to activate your P53 gene if it's not working. Maybe you'll take a pill just like we take vaccinations right now. Maybe we'll have cancer vaccinations for the most common cancers as well. And let's look at our questions. Okay, so this is, I don't really like this question because I can understand why it'd be confusing, but the idea here is that a proto-oncogene could cause cancer if it became a what?
An oncogene.
If it became an oncogene, then it would cause cancer. Which of the following are potential causes of cancer? Carcinogens, yes, yes, no.
Yes. Yes. No. Yes. Yes.
Okay. And then, which of the following are ways to diagnose?
Yes. 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 Yes.
And no. Okay. All right, and then that is the end. Okay. Yay! That's your last chapter for the semester. Okay? If you're staying up late, have a piece of toast, make good choices. Missed you.